Thanks. So I'm Jill, and I'm a postdoc in Heather's lab, and I'm new to ACTA2, so I'm really happy to be here. So I don't think I really need to go through this slide, but I can tell you that I'm like pretty new to this disease. So I'm also work on other projects that are rare diseases, and I think that they're really important and very devastating and need our help. And so our kind of lab and our team at UMass is really geared towards making gene therapies for rare diseases. And so she was here to develop a sheep model of MSDS syndrome. And so it's caused by mutations in the ACTA2 gene. This gene encodes alpha smooth muscle actin isotope, which is important for smooth muscle function and contractibility, which affects basically every single tissue within the body. It's inherited dominantly, and it is ultimately a fatal disease with only palliative treatment, and the burden on quality of life is pretty high, as some of the other presentations have already highlighted today. So we're going to have like two parts of this talk here. So the first part is the work that we're doing to develop a sheep model of MSMDS. And then also I can talk to everyone about sort of the path to the clinic. And Heather, who had to step out for a minute, will probably be able to answer more questions about the second part of this talk. And so the first, I will talk about how we're generating the sheep model. So why large animals, right? We kind of know that large animals better recapitulate human disease in comparison to smaller order animals. So, I mean, if we just look at this picture of all the different brains from all the different diseases or species, right? You can kind of see that the human is pretty big, different shape than, you know, a rat or a cat. And we're kind of focusing on sheep, right? So they're larger, more similar to us, and they tend to be really useful in determining therapeutic efficacy for different types of therapeutics. And so the question I always get asked is, why use sheep? So like I said, they're similar size, anatomy and immune systems to human. Again, there's like the mouse, sheep, and then the human brain. You can see the comparison, I think, just by eye. They're really easy and docile in nature. They're smaller, more manageable in size, unlike pigs and cows, which our lab also works with. And if anybody works with mice, you know that they're kind of really not nice either. They tend to bite a lot and they do hurt. They're not as controversial as companion animals. I have 2 dogs at home. Those are my dogs, right? So finding researchers that are also able to and willing to work with certain research animals is also something important to consider when you're making large animal models. So less people want to work with animals that are pets in your home, livestock animals, right? Most of us consume them as meat, so there's a little bit more of a disconnect there. And of course, they're safer and more ethical and less expensive than our closest living relatives, the non-human primates. So hopefully I convinced you why sheep are so great. They're also very cute. So our methods for generating large animals, there's a bunch of different ways that we can sort of tackle this. So the canonical way is doing the somatic cell nuclear transfer. That's how Dolly the sheep was born. And so basically you kind of take a cell, you modify it, and then you introduce it into the egg cell, and then you kind of like shock it and then make it an embryo, and then that holds your mutation or your variant that you create. You can also do this by microinjection, so you can insert things inside the embryo by actually injecting into it. And then our method that we're using is called electroporation. So basically we're just shocking components outside of the embryo and then hopefully getting them inside by making little holes in the membrane of the embryo wall. And so in generating a sheep model of MSMDS, we're using a DNA donor template. And we use this with CRISPR-Cas9. So this is an enzyme that can make a double-stranded break within the DNA. So we use this with a guide RNA that's targeted to a specific region of the DNA. CRISPR comes down and makes a cut based on where the guide lies, and so you get a mutation. But we can also incorporate foreign DNA to be inserted into the DNA or incorporated. And you can do this a couple of different ways. We've tried a couple of different strategies. So we try to do double-stranded donor DNA, but that unfortunately didn't work, as she face there. And then what we ended up finding to be most effective is using a DNA donor that's then put inside an AAV, so an adeno-associated virus. And then we inject this into the cell. with our Cas9 and our guide, and we electroporate that all together to then hopefully have insertion of this variant, which we're using the R179H variant that's pathogenic, and we've kind of talked about that today as well. And so we made a donor sequence that contains this humanized R179 amino acid change or pathogenic variant. So the little blue dot, which some of you might be hard to see, is where the mutation is. So it's a G to an A. And then we've tested a bunch of different guides, but because we don't want the Cas9 to kind of keep cutting, we then silence what's called the PAM site, which is where the Cas9 kind of binds so that it doesn't keep cutting. So our DNA construct is humanized, but also silences those other nucleotides so that the Cas9 doesn't keep cutting. So looking at the sequence homology, we looked at pig, human, and sheep and aligned them. We then designed guides to kind of be closer to where we want the mutation to be. And then how we do this in the lab is we screen these different guides in our sheep fibroblasts that we isolated from our sheep flock. We do initial testing just to see if our electroporation protocol works with a GFP reporter. How many cells turn green? Is it effective? And then what we do is we test the guides. We then isolate the DNA, and then we look for indels or mutations within the sequence or within the sheep fibroblasts, and also looking for incorporation of this mutation. So for right now, we're just looking at guide screening, so which guides work the best. And just to orient you to the graph, so the little blue dot there is the mutation, and then we have our two guides on either side. And for lack of a better scientific word, right? So you can see these are chromatographs, and they have these little peaks. And you can see the one that's kind of in the second one down looks really clean. So that guide didn't work really well. And if you look at the guide from 10854, those sequences kind of look really jumbled and messy. And that means that the guide works. So that was great. We found a guide that works really well in our sheep fibroblasts, which will translate to the sheep embryo. And so then we moved into sheep embryo editing, right? So we screened them in the fibroblasts, and now we're going to go into the actual sheep embryos. And how we do this is we flush early stage embryos from sheep. We then microinject these embryos with our AAV6 that contains our donor template, the R179H. We then electroporate our Cas9 and our guide with embryos that were microinjected. 24 hours after they've been microinjected, we then let them grow to blastocysts, some more cells to then do the analysis. And then we look for incorporation of our donor called homology-directed repair. And so our first kind of set of experiments, we had a lot of sequences that were homozygous, which we believe to be incompatible with life, right? We know that ACTA2 is a dominant mutation. And so you can see that there's mostly A's there. There weren't really many G's, so that's the mutation. And there were also a large amount of normal embryos as well. So we decided, because there was mostly homozygous, maybe we're just injecting too much vector, so we kind of diluted the vector down, so less of the DNA donor template that's being put into the embryos, but we still saw pretty similar results with most of them being homozygous. So then we decided that we were going to include both a wild type ACTA2 gene along with the mutant ACTA2 gene. And so these are in separate vector constructs, but they're both packaged into AAV6. And then we injected them at a ratio of 50-50 into the embryos, and we have an increase of survival of our embryos, so around almost 70%. Most of them are heterozygous, which is good for us. And we're around the 50 to 40% range for our insertion of the mutation. And I will say, the highest percentage of reads that actually had our mutation were 40%, and that cell actually halted at a 32 cell stage. I think kind of indicative that having more homozygous reads is probably pretty detrimental to the embryo, right? It seems to be important. So we found the most efficient delivery system to deliver our DNA donor construct. Our first kind of experiments with the mutant donor alone was mostly homozygous embryos. We diluted it, and then again, still mostly homozygous. We included the wild type ACTA2 donor, and then most of those reads for each embryo were heterozygous, so that's pretty great. And in another separate project that I did as my thesis, was developing another sheep model of a different rare disease. I'm testing different reagents and chemicals to kind of improve the homology-directed repair or the incorporation of that variant into the embryos. And so the green is their survival rate, and then the black is the percentage of HDR, which means the percentage of reads that do have the incorporation of our mutant. And this again is for a different project. But once this is done, we hope to then pick whichever compound is best and use that with the ACTA2 donor, both mutant and wild type. And then hopefully we want to implant some of these HDR positive embryos so we can generate an actual sheep model of the disease. And so the next part of the talk is I'm going to talk about the path to the clinic. So we're working with Patty at MGH. So basically, getting the path to the clinic is we start with proof of concept studies in animal models, and that's usually done in-house. They have done that, and so then we talk to the FDA, and because they're using a base editor, which is another way of editing the genome. The FDA came back and wanted us to then test this in a larger order animal, so we're going to use our sheep. But we're going to look at, they're going to be normal sheep, so we'll inject them and then look at off-targeting within the sheep. So let's just say that we had a lot of distribution to the liver, right? Then we're going to have to look at those off-target assessment in human primary cells. So sheep and humans are different, our genomes are slightly different, so we want to make sure that what we give to patients is safe. So the FDA has recommended us to do that. We then do a pharmacology and toxicology study, it goes to product manufacturing, and then hopefully we'll get to a clinical trial. So we're already kind of on this path. And so we're going to inject 2 lambs pretty early at birth, so probably within the first few days of life. We're going to give them around 8 E13 vector genomes per kilogram, similar to the one that's going to be for patients. And there's 2 different vectors because the cargo is a little bit too big to fit in one EEV. We're going to monitor them for health and then euthanize them 1 month later, harvest the tissues, and then look for presence of the AAV vectors within these tissues, and then look for the editing of sheep ACTA2 locus, even though it will be different than humans, but it will give us kind of an idea of where to look for off-target events and how effective the base editor is. And then the next step is to edit primary human cells of the tissues of interest discovered from the lamb study, as I said. And then right now our status is AAV manufacturing is kind of undergoing right now. And we have lambs that are gestating, so we plan to start this in the early fall. So it is getting going. And again, if you have more questions about the second half, Heather can probably answer those. But this is a very collaborative project. So again, I thank my lab and particularly Abby, who was in the lab, who kind of started this project and then left. So it's now in my hands. The Scott Wolf lab, the TAMCOR lab, who has done most of the embryo work. The Tufts Veterinary School, who houses all of our sheep. Mass General Hospital, so Patty and Ben. And then, of course, our vendor resources. And I'm happy to take any questions.
Great that you're developing a large animal model. What would the FDA say about the on-target effects? What's acceptable and what's not?
That's a good question.
It's a conversation, isn't it?
Yeah, Heather might be able to speak to more about that. She deals with it.
Or you can just pass it to Patty, because she's closer.
Yeah. Or yeah.
So the feedback has been it's a very challenging thing. You want to figure out what the biodistribution, which is basically a fancy word of saying what cells does the virus infect. Do you need to know What they want to know is what cells do the virus infect? So that's one question. And so that's easy for us to tell. In an animal, we can go and collect tissues and find out which tissues the virus liked best. And then of those tissues, they want to know what the editing was like. And so if you think about the genomes, there are similarities on the protein level, but when you get to the genetic level, it changes dramatically. And so an off-target effect, which is basically genome editing in places where you don't want it to be, okay? That off-target effect that you would find in a sheep would mean nothing to the off-target effect that you would have in a human, because the genomes are not even remotely close to the same thing. There, even if they have the same genes and have the same protein. So what we have learned, and I will say we, it's really Patty and her team more so than me, but what we have learned in this little journey is that they want to know what tissues are infected and what the ACTA2 editing looks like. Because the gene is slightly different in the region of interest, the drug that is being developed for humans will not work in the sheep, okay? So what Ben did was he designed a similar drug that also will edit ACTA2 in a different place. It doesn't tell you anything about efficacy per se, but will tell you the likelihood of the genome editor being able to get into that gene and make that edit in that tissue in the environment in which it needs to be, like a human. Once you've figured out what the editing of that gene of ACTA2 is in a sheep, then you can go to human cells, put the virus in human cells, and then see where that genome editor cuts. That's not appropriate. The edit's not appropriate. Because that's actually at the end of the day what you're looking to find out, right? Is what would be the changes that would happen in there. And so our team has kind of pushed pause on the sheep model and focused our resources on helping bring this really great therapy to the clinic. And my lab, and Jill specifically, has experience running toxicology studies to enable clinical trials for gene therapies. And so we're basically just doing whatever we can to help move this forward. And in this particular situation, it's this study trying to figure out in an animal that's on par with the size of a person, or as close to, not quite the same, but on the same order of magnitude, at least we'll say. Lamb's about the same size as a newborn child. It's not that much different, a couple kilograms. Yeah. We will have an idea of what dose we need to use to get to the amount of editing that we want, and then from there we'll enable the data to be collected in human tissue. Sorry, that was a long-winded answer, but did that make sense to everybody?
Yeah. Yeah, and I would say, as Heather was emphasizing, the only off target of the genome part is always human cells. The thing is that they want to see relevant human cells, and that has to come from which ones are going to get edited. So all this effort is, we're not only thinking about ACTA2 R179, we're working on several programs for other genetic vasculopathies. So everything we really develop at this level of knowledge will be very helpful to translate to the next one, the next one, the next one. So it's a really large effort. It's basically developing a full therapeutic for the lamb, which is not a therapy, it's just a pretend edit. It's very close to where we need to edit, so it's relevant. And we'll hope that that tells us what cells we really need to be looking into the human. Is it a liver cell plus a vessel cell plus kidney cell? We don't know exactly which ones are having significant levels of editing if we don't do this sheep. And just to give you an idea, all of this has been a process of 2 years of discussions back and forth between a large group of scientists and the FDA, and none of this is in the regulatory documents, except for the off-target, because they basically don't need this information if we cannot provide it. So they basically know we're doing it, but they are waiting and hoping that we can inform further how we do this. In short, for everybody that is not following the therapeutic stuff at the moment, because we haven't talked directly about the genome editing, we're trying to check everything we can in any technique and model that we have to make sure that it's the safest. The only concern here is it's safe to try it in a human. So that's all this effort. And I will thank you for bringing another career into MSMDS.
Happy to be here.
Yeah. Any other questions?
I see that the
what you've done so far is for the ACTA2 R179H variant. Are you guys also working on this for the other ACTA2 R179 variants like the R179C? And if not, do you have to start from the beginning all over again, like what you did with the 179H as far as getting like FDA approval to keep like small mouse study, FDA approval, move to the large animal, or would they be the other variants be grouped in?
Yeah, I was like, Patty might be able to get a better answer than question than me.
So, an amazing question.
Yeah,
Okay.
very good question, actually.
As you guys have seen, developing gene therapy takes a lot of work, a lot of resources, and a lot of years. And we know the disease in ACTA2 is aortic dissections in all variants, like 27, 158, 258, 179. You get aortic disease. And we're doing all this effort for one of them, RG179H. And the question is, will we be able to recycle any of this to do the other ones? So here's what's happening right now. In fact, yesterday there was a workshop at the FDA. It's publicly available and recorded if anyone wants to watch it, about rare diseases. And it was basically announced what the FDA has been working with us and other groups at University of Berkeley, UCSF, Hopkins for years now, UMass, which is we're hoping to submit this proof of concept. No, we do everything in the mouse, everything for RG179. And then in human cells, we show them that we can do the similar surrogate, like the amount of editing that we can do in human cells for the other variants is similar. We can't iterate too much on the enzyme, so it will be limited to what the enzyme that we are using can do. But yes, and it's not just for vascular, there are liver diseases and stuff. This is going on right now. The idea is that you get one trial approval, it's called an IND, and the first patients to be treated will be the ones with the mutation with all the package, but we will have in the package the other mutations. And ideally, were the funding to be there, you'll be able to move to the next cohort without having to do any of these several years of work, like more than you saw. Andy is ahead of us, and he's been working on this for 20 years. Now he's putting one therapy after another. Probably, I would say, I'll give you 3 to 5 for the next clinical trial. It takes much faster, because he paved the way for his particular model, clinical trial design, the delivery method. And now he can, again, use a lot of that know-how and the resources and the regulatory experience. And as soon as there is safety in humans, the FDA, the EMA, the UK, the regulators are just trying to prevent people from experimenting in humans with things that could be unjustified risk. Okay, that's their job. They want you to have, if you're going to touch a human, justify the risk you're putting them through. But I think this is happening, and it will be happening in the next 2 to 3 years. The FDA changing the way they allow us to do, specifically for genome editing, not all kinds of gene therapies, but for genome editing, iterate. You saw what Abby was showing. You can change the guide and move the enzyme into another part of the genome. That's what we want to do with the other mutations. So, amazing question.
Any other questions?
There's one in the chat. Megan in the chat said, how long are we looking for the trial on humans to start?
That's a great question.
Yeah, exactly. It's always you.
It's always
Yeah.
So, in this computer, we have the gram. Gonna give you a little bit of background, but we were hoping to have the authorization from the FDA if the studies in these large animals were showing favorable toxicity, meaning they were safe. I'm saying if, because this needs to be done, and it's not yet. So if those safety studies were okay, we were hoping to dose the first patient in 2026 at the end or beginning of 2027. But the funding to do these studies, as you saw also from Andy's presentation, is very heavy lifting. They're very expensive things to do. And it was recommended by the NIH in December. It was supposed to start in June 30th, in 3 weeks from now. We did this application with Dr. Lindsay and the whole team back in February last year. But there's been changes in policy. They cancel everything and they basically ask us to now submit it again today, by the way, before 1 hour from now. To be able to fund it with a different resource, because the resource they had already recommended for funding that they were doing with us got axed by 40%. So they couldn't do it any longer. They're still trying to help us, but we don't know if we're going to have those resources as we were expecting from the NIH, but we're doing everything we can. So that's what marks the timeline. So we wanted to be in first in human in latest first quarter of 2027, but the funds that were supposed to come in 3 weeks will not appear until at least February next year. So right there, you have 9 months extra. And then we'll have to see, because there might not be resources or not. It's a very uncertain time. I don't want the drama or the depression or anything, but it's real that the rare disease community is being affected in a much more scalable manner than maybe others. I would say everybody's affected because diseases affect common diseases, and chronic diseases are also being damaged by some of these policies and cuts in funding that already happened. So we're in it, and we will also try to fundraise whichever other way. It's not just has to be the federal government in the US. Andy put us in front of a bunch of different resources to be able to apply to the UK. They have done gene therapy in vascular in children, both in London and in Edinburgh. It's possible that we have to pivot, and we'll pivot. We keep pivoting every 5 minutes. So that's what we do in the lab. So, and the clinic too.
So that just goes to the point that we made yesterday. I'm not sure if you were online at the time, Dr. Marcelino, but this is what we meant by the funding uncertainty and how it could impact us. There's a very tangible impact that she just articulated for us. So just another plug to help assist in the fundraising there. We did have another follow-up question from that individual asking for you to specify how much funding is your shortfall currently.
You really want to know?
How much money do you need?
The full execution of the studies that are needed to get to the authorization for first in human and the clinical trial with the first 6 patients dosed is right now the budget that we put is around $11 million. And we were able, this is why I welcome the crisis, we were able to lower this cost from 20 to 11 because of these changes right now. So we have to work, and we found an amazing team that could work with us at much lower cost than NIH could find. So there's benefits and profits in the crisis, I think, but that's what we need for genome editing. We're also developing a small molecule. I want to put a shout to the Belanger Family Foundation. They have been funding the small molecule development. That's much closer right now to clinic. I just had on Monday in DC conversations with Biomarine, seeing if they can provide a drug for free. That study, the effect is modest. We'll be presenting some of that tomorrow. It's really good, but it's different than genome editing, but I think we have to do it. Not everybody will be qualified for genome editing or gene therapy, and we don't even know if it works. So I want to be very open that all therapies development in our group or any group are good for the disease. And the other one is we're doing an RNA therapeutic as well. So for the small molecule, we need the run to the clinic now. We think with the Belanger Foundation, we are completing the preclinical work. So now we need to run to the clinic. That's below $2 million. And then the small molecule, to get all the way to the clinic, we'll need around 3. So these are pick your items. That's the fundraising. And for everything I'm talking about, we have multiple submissions to different sources of funding in Europe, in the US, with industry, and with philanthropic support always, which gets us to keep moving when there are times of uncertainty. The last thing we want is to lose an Abby or a Heather Lab or someone like that, because you can't recreate the expertise in a blink of an eye. It would take years to learn. And you've seen, like maybe in the presentation, all these data and troubleshooting, years and years of work. Let's not lose it. it will be a delay for us to get to clinic or to get to insights in the disease also that could help another disease, not just MSMDS.
Thank you.